Welcome everybody to the second of my talks on variation in the human genome. This time, how can we make sense of it? So, understanding variation, it's important to remember that the average healthy human person has about 100 loss of function mutations. And this brings a real challenge to interpreting whole exome sequencing data. If we find a loss of function mutation, we might find 100, and we have to try and figure out which of those could be contributing to the phenotype that we're interested in. And again, population studies recently have shown, quite surprisingly, that even healthy humans have knockout mutations, by which we mean a complete loss of function in 2 copies of a gene that is predicted to have a pathological effect, and yet these humans are apparently healthy. So this has brought about a need to reconsider some of our pathogenic predictions. So human knockouts, as I've said, are very interesting. Healthy human people have on average about 15 complete knockout events. So it's important to remember that just because you find a homozygous or a compound heterozygous loss of function event, it doesn't mean that that event has caused the phenotype that you're studying. And if we look at populations with a high rate of parental relatedness, we find even more knockout events. So here in this population of an Asian heritage, about 1,300 genes showed knockout events, at least one in each participant. So these human knockouts provide opportunities for unique insight into medical genetics, unique opportunity to understand genetic effects. So we've already learned that whole exome sequencing provides information on variants in the protein coding genes. And when we find these variants, each variant requires functional assessment to understand whether that variation is actually having an effect on the protein and the phenotype. Similarly, genome sequencing and genome-wide association studies give information about intergenic variants or variation between the genes. And here again, we need careful functional assessment, this time often even more challenging than understanding variation within genes. So common variants, as I introduced in the previous talk, often cause common or mild disease. But it's important to realise that even common variants can contribute to rare or severe diseases. And this has been most eloquently illustrated in the field of neurodevelopmental disorders. So here it's been shown that multiple common variants coming together in a kind of a dose effect or contributing additive effects can lead to rare and severe phenotypes. So one gene doesn't necessarily cause one phenotype. We know that loss of function variants may be tolerated, and this tolerance can be a context-dependent effect. And by context, I mean the genetic context, so what genetic variation is present around that variant that you're studying in other genes nearby or within the same gene, but also the environmental context. So a specific genetic effect might not have a phenotype unless that gene comes into the context of environmental stress, in which case the phenotype might be expressed. And there are interesting links between common complex traits and monogenic or Mendelian disease. And the examples that I've listed here are just 3 examples of these. So we know that loss of function variants in the filaggrin gene cause the common monogenic disease ichthyosis vulgaris or dry skin, but they also contribute to the common complex trait of atopic eczema. Similarly, we know that variants within the CARD11 and CARD14 genes can increase the risk of eczema as a common complex trait, but loss of function or knockouts of these genes can cause a much more severe atopic eczema phenotype. Similarly, with DNase 1L3 and SLE. So how do we set about understanding which variants are likely to be important in disease or in normal human variation? So the key here really is what genetic variation results in a change in transcriptome that then results in a change in the proteome, because ultimately it's the proteins that are the building blocks of cells, and cells are the building blocks of our phenotypes and our diseases. And it's important to remember that we often study DNA as if it was a linear molecule, and because, you know, that's a simpler way to understand the sequence. But DNA is by no means a linear molecule. In every cell in our body, DNA is wrapped around histones, very tightly packed together, and then packed again in the 3D conformation that densely compacts the DNA to form chromosomes. So whilst we might study it in a linear way, trying to predict how different genetic regions might interact with each other, we need to remember that these genetic regions come together in a very complex 3D effect, and that can bring together different areas of DNA that appear widely distant in a linear model, but are actually very close by in the real 3D shape in vivo. So remember, we're interested in variation that changes transcriptome, that affects the proteome. And we can start to get a handle on what variation might be having an effect on the phenotype in various ways. So population data is important. If you find a variant that you think is contributing to a rare disease, and you look in the population and you find that variant is very common, then it's unlikely that that specific variant is causing a rare disease, given that the variant is common in the population. Conversely, if the variant is rare in the population, then it is a candidate at least for rare disease. There are many pathological prediction tools now that are available, and I'll talk through a couple of these in my next slides. So these are often in silico or computer-generated tools that predict effects of changes in DNA sequence or changes in protein on how likely that is to have a pathological effect. And then moving into a next layer of complexity, we can use in vitro models. So we can model changes in DNA and the effects on cells or tissues. And again, sampling skin, we're at a great advantage that we can model genetic changes in our cells and tissue of interest and actually see the effects. And then another layer of complexity is modelling effects in vivo. And here mouse models and zebrafish models and other organisms can be used to gain more detailed functional effects. So I mentioned already there are publicly available data sets that can help. And here are just some that I've listed, some of my favourites. There are many others. There are also publicly available resources giving information on gene expression levels and the functional assessments that I've spoken about. And again, I've listed quite a few here. These are not exhaustive lists, these are just some of my favourite resources. And I'm going to take you through a few in the next few talks. So 4 examples from my favourites. The ENCODE project, I've already shown you this diagram. The ENCODE project is an enormous wealth of data from international work that allows the sharing of detailed information about the control of gene function and 3D effects and intergenic regions and predictions of effects from gene sequence to transcriptome analysis. So this is a resource that's freely available online, and many happy hours can be spent clicking on different boxes and finding an enormous goldmine of information. Another example I wanted to share with you is a similar goldmine. So this time Ensembl, I've given you the link there. Again, all freely available. So you can choose your species of interest. You can look at gene expression in multiple different tissues. You can retrieve gene sequences if you want to study one specific gene or understand your gene in context. And this tool is useful, the variant effect predictor. So if you click on the variant effect predictor, it brings you to this page, and then you click on the launch VEP. And here you can put in variants that you're interested in. So either the sequence that you're interested in or a specific single nucleotide polymorphism, as I've put in here with an RS number. And then you just hit run the instant VEP to find out the predicted effect of that variant. And it often takes just a few seconds to run, and then the results are available to you. So here are just some predicted effects within my gene, my favourite gene, filaggrin. So you can see that the consequences are categorised as either a variant, a synonymous variant not having a coding effect, a missense variant, or an intronic variant. And again, you know, this is a freely available resource that you can spend valuable time searching and learning more about the variants that you're interested in or the gene that you're interested in. So another more recent publicly available resource that's been released is this GnomAD resource. And here again, publicly available, you can search by any gene, any region, or any variant that you're interested in. So here's my favourite gene, filaggrin. You type this into GnomAD, and one interesting aspect that you can find out is the predicted kind of likelihood of pathogenicity. So it gives you a prediction score of how likely the variants are to cause damage on a phenotype effect. And here you see filaggrin's an interesting gene. It has more missense variants than expected, and the predicted pathogenic effect is very, very low. So I think this is my last example, the Human Protein Atlas. So here, another publicly available resource, put in your gene of interest, which again, you're going to hear about later in the course, and just hit search. And you can find out so much about this gene. You can find out the full name of it, which tissues it's expressed in, both at RNA and protein level. And you can drill down on these tissues and see in a lot more detail the regions within the body, but also within the tissue where the gene is likely to be expressed on a protein level. And just to finish up then, I thought it'd be helpful if you're trying to address the question, is this variant important? I'll give you an example from recent dermatology literature. So this was a paper published by the McGrath group in London. And here they took a collection of patients that they found with hypertrichosis and loose anagen. And in one of the patients, they found compound heterozygous loss of function mutations in this gene TKFC. And so to define whether these variants were important, they really followed this complex pathway. So starting with exome sequencing that had defined these compound heterozygote mutations in this one family, this one trio was present in the affected child but not in her parents. The next step was to search in population databases, as I've said, to test whether these variants were common or rare in the population. And they were found to be relatively rare, consistent with a rare disease. And then trying to understand whether these variants might have an effect on protein structure. The group used variant effect predictors and actually went on to synthesize proteins with these variants within them to show that these variants then also had an effect on enzyme function, modeling in vitro. So really very complex, detailed work. And this complex pathway is necessary before you can really begin to apply genetic knowledge to therapy development. We've got to prove that this is likely to be a functional mechanism before we can start targeting it for therapy. So a challenging pathway, but surely potentially very fruitful in terms of translating for clinical benefit. So to summarize, there are multiple sources and mechanisms of variation within the human genome. Recent technological advances have expanded our understanding, but there's still lots to learn. There's loads of freely available data that's, impressively shared online. And skin offers unique opportunities for insight and also clinical impact and translation for our patients' benefits. So just to finish now with some quick recap questions for the most important points. So how much of your genome is protein coding? Is it 0.1%, 1%, 10%, 15 or 50%? Can you remember? So remember from this apple, the protein coding or exome is about 1 to 1.5% of the whole genome. Question 2, which of the following mechanisms contribute to variation between different parts of your own skin, so different regions within your own skin. And for this, you can answer true or false to each. So mosaicism, does this contribute to variation between different regions of your own skin? True, it does. What about germline mutation? Does that contribute to different regions within your own skin? No, that's false. So germline mutation inherited from your parents should contribute to the phenotype affecting your whole skin. Obviously, there are regional differences, but shouldn't cause distinct regions that are different. Conversely, somatic mutation. Now, this can cause different regions that are different within your skin. And epigenetic effects, can that contribute to variation between different regions of your own skin? Yes, that's true. And again, you'll learn much more about these later on in the course. Question 3. Which of the following is not an example of structural variation? Is it A, copy number variation, B, single nucleotide variant, C, chromathripsis, or D, deletion? Or E, inversion, which is not an example of structural variation? So hopefully you all got that. It's single nucleotide variation. All the others are different types of structural variation. And last question, question 4. Which of the following sources contribute to variation between people in this room? Sadly not in the same room, so here's a picture of lots of smiley people. A, loss of function variants, B, missense variants, C, regulatory variants, D, environmental effects. Which contribute to variation between people? Or is it E, all of the above? And of course it's E, all of the above. There's many different sources of variation between all of us. So those are my questions. I'm happy to take questions from you, either in the next session where we've got Q&A that's live, or you're welcome to email me. I'm happy to get emails and always happy to talk about genetics. So thanks for listening.